Okay, Dr. Mancha, take it over.
Okay, well, thank you everybody for joining us for this talk on epigenetics and Darwin. And this is just my bio slide there. I went to Florida State for my undergraduate degree in biology and UAB for my degree in medicine. Excuse me. I'm a board certified family practitioner. I did clinical research as well for about 15 years. And the bottom right article is one of the ones that I helped contribute to. This is the title of my talk. It's called Epigenetics, or Does Darwin Need a Rethink? And on the right, this is a quick overview of the difference between the theory of evolution and epigenetics and why they're different. If you look at the portion that says underneath the theory of evolution, you see the DNA helix and two double red lines. And then below it, you see a yellow gold star, and that represents a mutation in the DNA. And its evolution, its theory depends upon those mutations in the DNA to change the phenotype on the bottom. The phenotype is the outward expression of the DNA or the organism. Whereas epigenetics on the top right takes the DNA and it puts a purple tag, if you will, and it sits on top of the DNA. That's why it's called epigenetics, epi meaning above in the Greek. And in this case, that purple tag represents a few different things, including DNA methylation, non-coding RNA, that's junk RNA, and histone modification, which we'll describe later. And it causes a change in the phenotype at least 100,000 times as fast as the mutation process. This is what got me started, this article in Nature Journal, which was 9 years ago. And when I saw it, I couldn't believe it, because Nature Journal is like the journal of evolution and biology. And they basically realized that something needs to change. And they wrote this title, Does Evolutionary Theory Need a Rethink? Which I basically copied. In the article, they discussed that there was two different points of view. One point of view says, no, we don't need to change. And the other said, yes, we do, and everybody's joining us. And then they said, yet the mere mention of epigenetics often evokes an emotional, even hostile reaction amongst evolutionary biologists, perhaps haunted by the specter of intelligent design. Now, where'd they come up with that? Why did they even bring that in? Well, I mean, these guys they're afraid of that intelligent design is going to get them, but now epigenetics looks like intelligent design is basically what he's saying. Evolutionary biologists wish to show a united front, and that united front is basically the support of neo-Darwinism. However, one of the main things that epigenetics does is it brings teleology back into the mix. And teleology is the explanation of the phenomenon in terms of the purpose. So we got phenomenon and purpose rather than the cause. According to neo-Darwinism, the cause is mutation, and then there is no such thing as a phenomenon. From a theological point of view, it's called the doctrine of design and purpose in the material world. So this is basically getting down to the design model, which RTB supports. As you know, it's close to being Darwin's birthday. He wrote his Origins of the Species in 1859. I can always remember that because it's exactly 100 years to the day when I was born. And so you add on another 62 for me. So it's been 162 years since it's been written. This paragraph in the Origin of Species, which is free, by the way, you can download this off of Google Play. This paragraph really summarizes his whole theory, really. He says, and what's interesting is a lot of people don't realize what I'm about to tell you. He says, if the conditions of life. Now, the conditions of life was a technical term that he borrowed from Jean-Baptiste Lamarck, 50 years his senior. He came up with the conditions of existence. But he says that the conditions of life, which Lamarck was talking about, and Lamarck said this is the environment. The conditions of life is the environment that's around us that changes our traits. And Darwin goes to say, if those things change and the uniform undergoes modification, Uniformity of character can be to the modified offspring solely by natural selection, preserving similar favorable variations. And that's out of his Origin of the Species. This phrase is actually pretty close to being correct if you take into account the conditions of life part in Lamarck. But they don't. The neo-Darwinists chop out, they ignore the conditions of life aspect, and they just jump down to the natural selection. As I said, Lamarck was 50 years his senior, and Lamarck said traits we acquire during our lives are passed on. We call that soft inheritance. Darwin believed in this, but the neo-Darwinists were specifically against it. Not only the neo-Darwinists, but also the modern synthesis, which came along later. They specifically went out of their way to destroy, if you will, this concept of soft inheritance. And they won out for 100 years until epigenetics. Following the Origin of Species, Darwin spent much of the rest of his life searching for possible mechanisms which would communicate information from the environment, the conditions of life, to the genome. Of course, he didn't know what a genome was. He stated that there were two forces in evolution, natural selection and conditions of existence, the Lamarckism. Of the two, he claims that the latter is the more powerful. I mean, I couldn't believe when I read this article, because, he's basically saying natural selection falls second to conditions of life, and conditions of life equate with epigenetics, in fact. So in doing so, he recognized that natural selection could only operate within the bounds of possibility, that is the environment. And that's Lamarck's idea of the environment. And this article is called Darwin's Passionate Environmentalism, or, and this is an interesting part of this title, the dangerous fallacy of the all-sufficiency of natural selection theory. And he's basically explaining that neo-Darwinism was such a dangerous fallacy because they completely were in on natural selection as being the only thing going on, and they ignored the conditions of life. Conrad Waddington, who's the father of epigenetics, he proposed it in 1940, by the way, agreed with Lamarck's central ideas. Waddington felt Lamarck had been abused and ignored for 120 years by neo-Darwinists. Also, at the same time, in 1942, Julian Huxley proposed the modern synthesis. This is the revamp of neo-Darwinism. Neo-Darwinism was proposed in 1900, and then the modern synthesis incorporated neo-Darwinism. If you say neo-Darwinism today, everyone interprets it as being the modern synthesis. This is, quote, the theory of evolution. And he published in his book called The Modern Synthesis. The Modern Synthesis combines Darwin's theory of evolution and Mendel, Gregory Mendel, who you've heard about before, on heredity, along with distinguishing micro and macro evolution. I think I accidentally deleted a slide, but at the same year that this theory came out, Conrad Waddington wrote this article, wrote a long letter to Nature Journal, the major journal. And he said, and I'll paraphrase, he said, it's very unlikely that even the most statistically minded biologist is going to be happy just counting mutations along the natural selective. So basically, he was questioning that natural selection and mutations could really account for things. And his early experiments showed that it was outside of that. This is an illustration of the modern synthesis. It includes the natural selection in the middle on the left and survival of the fittest. And genetic variation in the middle was caused by mutations in the top right. So you have your mutations causing genetic variation, and then you had the Mendelian inheritance on the far right. This is something I showed last talk about the central dogma, and it's all since the modern synthesis came out in 1942, the central dogma was proposed in the 1950s by Francis Crick, the discoverer of the DNA molecule, and this, in essence, became the theory of evolution. And it ties in the modern synthesis with DNA, and we have the DNA, which then transcribes to RNA and then to ribosome. However, this is only for 2% of the DNA, which codes for protein. There's another 98% that codes for non-coding RNA, which is from junk DNA. So epigenetics is the study of how your behaviors and environment can cause changes that affect the way your genes work. Unlike genetic changes, epigenetic changes are reversible, and they do not change your DNA sequence, but they can change how your body reads a DNA sequence. And this is the key difference between the two. And this was from the CDC in their official statement about it. Again, let's go back to that very first slide that I showed. On the far left is the theory of evolution, and it depends upon the mutations, which were part of the modern synthesis. That being a gold star, and it changes the DNA sequence, and that causes a change in the phenotype or the outward form. Whereas epigenetics sits on top of the DNA, and different mechanisms work to change how the DNA is played. So epigenetics plays the DNA. It doesn't directly change the letters of the DNA, except in rare circumstances. I'll give y'all a choice. There's a TED talk, which is a little bit more simplistic, and then there's another talk that I have here, which has a little bit more gravitas to it. You want to go with the second one?
Yes, a TED talk. I'm impressed.
You like the TED talk? Okay.
Yeah, yeah.
What's a TED talk?
Here's a conundrum. Identical twins originate from the same DNA, so how can they turn out so different, even in traits that have a significant genetic component? For instance, why might one twin get heart disease at 55 while her sister runs marathons in perfect health? Nature versus nurture has a lot to do with it, but a deeper related answer can be found within something called epigenetics. That's the study of how DNA interacts with the multitude of smaller molecules found within cells, which can activate and deactivate genes. If you think of DNA as a recipe book, those molecules are largely what determine what gets cooked when. They aren't making any conscious choices themselves. Rather, their presence and concentration within cells makes the difference. So how does that work? Genes in DNA are expressed when they're read and transcribed into RNA, which is translated into proteins by structures called ribosomes. And proteins are much of what determines a cell's characteristics and function. Epigenetic changes can boost or interfere with the transcription of specific genes. The most common way interference happens is that DNA, or the proteins it's wrapped around, gets labeled with small chemical tags. The set of all of the chemical tags that are attached to the genome of a given cell is called the epigenome. Some of these, like a methyl group, inhibit gene expression by derailing the cellular transcription machinery or causing the DNA to coil more tightly, making it inaccessible. The gene is still there, but it's silent. Boosting transcription is essentially the opposite. Some chemical tags will unwind the DNA, making it easier to transcribe, which ramps up production of the associated protein. Epigenetic changes can survive cell division, which means that they could affect an organism for its entire life. Sometimes that's a good thing. Epigenetic changes are part of normal development. The cells in an embryo start with one master genome. As the cells divide, some genes are activated and others inhibited. Over time, through this epigenetic reprogramming, some cells develop into heart cells and others into liver cells. Each of the approximately 200 cell types in your body has essentially the same genome, but its own distinct epigenome. The epigenome also mediates a lifelong dialogue between genes and the environment. The chemical tags that turn genes on and off can be influenced by factors including diet, chemical exposure, and medication. The resulting epigenetic changes can eventually lead to disease if, for example, they turn off a gene that makes a tumor-suppressing protein. Environmentally induced epigenetic changes are part of the reason why genetically identical twins can grow up to have very different lives. As twins get older, their epigenomes diverge, affecting the way they age and their susceptibility to disease. Even social experiences can cause epigenetic changes. In one famous experiment, when mother rats weren't attentive enough to their pups, genes in the babies that help them manage stress were methylated and turned off. And it might not stop with that generation. Most epigenetic marks are erased when egg and sperm cells are formed. But now, researchers think that some of those imprints survive, passing those epigenetic traits on to the next generation. Your mother's or your father's experiences as a child or choices as adults could actually shape your own epigenome. But even though epigenetic changes are sticky, they're not necessarily permanent. A balanced lifestyle that includes a healthy diet, exercise, and avoiding exposure to contaminants may, in the long run, create a healthy epigenome. It's an exciting time to be studying this. Scientists are just beginning to understand how epigenetics could explain mechanisms of human development and aging, as well as the origins of cancer, heart disease, mental illness, addiction, and many other conditions. Meanwhile, new genome editing techniques are making it much easier to identify which epigenetic changes really matter for health and disease. Once we understand how our epigenome influences us, we might be able to influence it too.
Hey, Dr. Mancha, how long is the
Yeah.
other tape going to take?
It's about 4 minutes.
Can you play that one too?
Yeah, sure. In fact, why don't I just play those and go home because they do the whole thing for me. Can y'all see this thing that's popping up on my screen here?
Yes.
I don't know if doing this.
Hi, it's Mr. Anderson, and in this video I'm going to talk about epigenetics. In science, there's this age-old question, are you the way you are because of nature or nurture? In other words, am I the way I am because of the genes I inherited from my parents or from the experiences I have during my life?
Excuse me.
And a great place to study this is using twins. And so if I had an identical twin, we would have exactly the same DNA. But we wouldn't look exactly the same. And that's because
Okay, can you hear me all right?
Yes.
Let me get back to, I don't know what that happened there. Probably was pop-up ads. Let's see, we should be. Okay, and this is the next one. And this one's great. This was around 6 or 7 years ago. And it's really it's taking a second to load here.
Okay.
There are over 100,000 skin cells in 1 square centimeter of skin. Each of these cells contain close to 2 meters of DNA, which contain our genetic information. All the genetic material in our body is called a genome. Because our DNA is very long, cells package up their DNA together with proteins known as histones. The DNA and protein complex is known as chromatin. DNA is wrapped around histone proteins, forming repeated units of nucleosomes, then further to form a chromosome. We have 46 chromosomes in total, stored in the nucleus. The core of one nucleosome is composed of DNA, wrapped around a histone octamer that consists of 2 copies of the major types of histones, H2A, H2B, H3, and H4. This organized DNA protein complex allows the cells to regulate what genes are expressed and when. All the cells in our body contain the same DNA sequence. Skin cells, muscle cells, and liver cells contain the same DNA sequence. Yet all these cells have different structures and functions. The reason is that different cells only use or express certain genes. Within each cell, the DNA and also the histones can be tagged by tiny chemicals that modify gene expression. These chemical tags cause some genes to be turned on and some genes turned off. For example, muscle cells will have genes turned on that a muscle cell requires, genes that a liver cell requires. Similarly, the liver cell will have genes for muscles turned off and genes that a liver requires turned on. These modifications are known as epigenetic modifications. Epi is Greek for above the genes, so modifications above the genes or on the genes. Epigenetic modification brings about lasting changes in gene expression. Let's have a closer look. Here is a normal human cell, and we are pulling out its genetic material. The DNA is wrapped around nucleosomes, which are made up of histone proteins. Coming off the histone proteins, we have histone tails. DNA is made up of a combination of 4 nucleotides, G for guanine, C for cytosine, A for adenine, and T for thymine. There are 2 main epigenetic modifications. The first one we will talk about is DNA methylation, where a methyl group is added directly to a cytosine residue that exists in a cytosine guanine sequence, or CpG sequence for short. So for example, here we have many CpG sites that make up a CpG island. And here the cytosine are methylated. The methylation of CpG sites in promoter regions is associated with gene silencing. The second type of epigenetic modification is histone modifications. This is where acetyl or methyl groups are directly added onto histone tails, and this will modify gene expression. For example, histone 3K9 acetylation correlates with transcription activation, and histone 3K27 trimethylation with transcription repression. Genome-wide patterns of DNA and histone modifications, or epigenome, are established during early development and are maintained during cell division. In cancer, these patterns are altered and disrupted.
Okay, did first one or the second one?
Both very good.
Really? Yeah. Well, the first one is more basic, but they're really both kind of basic. But the first one is a little bit, So, okay. All right, we're back on the talk here. And I was going to kind of take a moment to go back over talking about the central dogma, which we had talked about almost ad nauseum at the last talk. But on the central dogma is on the far left, and it shows the DNA going to RNA, going to protein, and that represents 2% of the DNA, the protein coding DNA. And then on the left, right, you can see how the junk DNA fits in between the red coding sections, if you will, and it's called the non-coding RNA. Non-coding DNA. And this non-coding DNA makes for non-coding RNA. And this non-coding RNA goes out and changes the RNA and the regular RNA and the proteins. And we'll talk about this further. So this is an overview of what you just saw. You have your chromosome on the top left, and it's surrounded by chromatin. And these are proteins which tighten up the the DNA, and it includes epigenetic mechanisms. And then when you look at them closer, you get what's called the nucleosomes, which is made up of several histone balls, if you will, that look like basketballs. And they have tags on the tails that come out of them, and they can be can moderate things to turn on and turn off. And it can be quite sensitive. There's a rare cancer in pediatrics, brain cancer, that just because of that histone tail, that one tail, if you look at the red yellow illustration, just in the one tail, it's off, and it causes a brain cancer in the pons, which is critically, it's bad. And then you go around clockwise to the red tags. That's the DNA methylation seen on top of the cytosine residues. But then you go to the bottom, and it talks about RNA degradation, which is caused by microRNA. That's part of the junk DNA that goes to non-coding RNA, and it moderates how the messenger RNA works. If you, we have 40 trillion cells in our body. Each cell has six feet of DNA if you unravel it. If you put it end to end in space, it would go from the Earth to the sun and back 600 times. So that's just an incredible amount of information. It's more than all the computers in the world, all the code in our one body, in the DNA. And all of that is controlled and handled by the epigenetics, which wraps it up and makes it tight so that it will express or not express things. This is a diagram that's a little more complex. And it really is just a rehash of the second video we watched. In the top left, you have the DNA modifications, which are typically cytosine residues put on the little red tags put on top of the cytosine, which you can see. But it can also latch on to others, but mostly cytosine. The top right, you have the chromosome modifications, and that's the histones. Yo-yos looking things, and then they can put tags on the tails that it carries around, and that also moderates whether it tightens up or not. In the bottom left, you have the non-coding RNA, which comes from the junk DNA, and it interacts with the regular RNA to cause it to code correctly, as well as the protein on the bottom right. So the researchers estimate that epigenetic change rates are as fast as 10,000 to 100,000 times faster than a genetic mutation rate. So that means that our body, as it's changing to the environment, which can include everything from child abuse to you name it, to smoking cigarettes, it can change the way our DNA acts quickly, and it doesn't rely upon the slow mutation process of neodarwinism, which you see on the right. It shows you see the blue DNA sequence variations, and I put an X through it because it's really a minimal player at this point. Whereas on the left, you see the orange juice and kiwi and other fruits, which are good for epigenetic modifications, and they eventually affect your phenotype or your body. This is an interesting concept. On the right was an article that was talking about epigenetic aging. I was talking to a colleague the other day, and he said he heard that in the next 10 years, we may be living 140 years on average. And that is because there are these trials that are coming out with positive results, increasing the ages of mice and things. It's in some way, they're actually using some off-the-counter medicines to actually do this in combination. But you can see from the top right, you have fewer epigenetic tags relating to aging, and then you pick up aging epigenetic tags as you go down towards the bottom. And this could actually explain what happened in the Garden of Eden on the left. You have Adam and Eve ate of the tree, but they didn't die instantly. But when they tried to eat of the tree of life, they were banned by the cherubims. There were 4 of them. That's what caused them to age ultimately and die, is their lack of having access to that tree. So it raises the question: when they were in the garden, they were created very good, but they weren't necessarily perfect from a no lack of entropy. It may be that tree was just kept prolonging their lives while they were in the garden. That's complete speculation. This is an interesting couple here. You see on the left, an older couple been married to each other forever and are deeply in love with one another. They did a study actually of couples like that, and they discovered that these guys had the lowest amount of epigenetic age clock malfeasance. They've been able to dice out of our DNA the epigenetic marks that account that are close to or represent aging. And when they studied their epigenetic signatures, they found that they were lower based on the fact that they were married for a long time. And I found that very fascinating. So for Darwin, and this is really in Darwin, but neo-Darwin, for mutation to be passed on, it has to be in the seed. And this, I always kind of found this interesting when I was in college biology and evolution. Everything had to have happened in the seed. You know, some mutation doesn't have, you know, if you have a mutation in the liver, it's not passed to the seed. Therefore, anything that happens during your lifetime is not passed on. Epigenetics, however, allows life's experiences to be passed on in what's called soft inheritance, and they are typically transmitted to the third and the fourth generation. This is a tongue-in-cheek article that came out in Nature Journal, which I'm amazed that Nature Journal published this. In fact, you see Homer Simpson on a motorbike with Bart behind him, and you just know that Bart's going to be, you know, a chip off the old block because of Homer's bad behaviors. And they basically, the title is Epigenetics, the Sins of the Father. I mean, they lifted that right out of Exodus 20. And you know that there are some researchers that must have totally grit their teeth when they saw this. But when they started studying mice and other things, and like that first film showed where they did a mice study and they withheld care, nurturing, and the child passed it on, it actually passed it on to third and fourth generation. And this is being noticed all throughout these different experiments, that they're going on to the third and the fourth generation. So it caught the attention of these researchers. This is Exodus 20. You shall not make for yourself an idol. You shall not worship them. For I, the Lord, and your God, is a very jealous God, visiting the iniquity of the fathers to the children, to the third and fourth generations of those who hate me. And, you know, I used to think that was very unfair, that the father's curse would actually land on him. But this is not, in essence, a curse like God's going to ding the great-grandson because he's related. This is a warning, in my view, of epigenetic trends. And he gives them the 10 Commandments right after that. And if you follow the 10 Commandments, you're going to not have those third and fourth generational curses because of the things that it tries to keep you out of. With epigenetics, the organism has the mechanisms to adapt. Now, this is the big thing, this adaptation, which is different than the word evolution. It's the ability to handle the environment. Evolution is mutations changing the DNA. Epigenetics is adapting to the environment. And it's largely without changes to the DNA letters. Again, looking back at something similar to happen in that second video, you have the octamer histone complex, and then see the stretch of DNA between them. As the little epigenetic tags get sit on top of it, they tighten up. And when they tighten up, they don't express DNA. The DNA is in a shutdown mode. And that's in essence where most of our DNA stands for. I mean, you know, remember the 600 times the sun and back? It can't be in a single strand. It needs to be tightened up. So we only play it at just the right times. So for 60 years, the neo-Darwinists felt that DNA mutations with natural selection caused evolution, but epigenetics mainly works without mutations. Even though epigenetics works without mutations, there still are mutations, however. However, epigenetics controls them as well. They are not random per evolution. This is an article that came out last year. It's a Nuclear Bomb Journal article that came out. And it says why mutations is not as random as we thought. And then it talks about the general idea is that mutations accumulate across the genome randomly. And so we would expect that, that acquisition of mutations would cause evolution. So they looked at mutations as being random. This is a slide I borrowed from Francis Collins, where he was explaining random mutations, but you can see the dice is random and it caused the mutation as an orange star. In the other article, the Nature folks go on, since the first half of the 20th century, evolutionary theory has been dominated by the idea that mutations occur randomly with respect to their consequences. We conclude that epigenetic mutational bias, this is the non-random epigenetic mutational bias, which I'll explain, challenges the prevailing paradigm that mutation is a directionless force in evolution. That is very important. It's, you know, before Darwinism and evolution was a directionless force. Now we're having mutational bias. The mutations are not really random, but they're challenged and they are biased in a certain fashion. So our discovery yields a new account of the forces driving the patterns of natural variation. So they're using the word natural variation, not natural selection, challenging a long-standing paradigm regarding the randomness of mutation. I mean, those two paragraphs are huge. They basically, you know, this is setting it up on, setting neo-Darwinism up on his ear, if you will. So, and again, using the diagram, they're loaded dice that cause bias changes, not random. For 160 years, the majority of evolutionists philosophically preferred the implication of a directionless force in nature as first proposed by Darwin. I mean, think about it. Philosophically, if it's just a directionless force, then we're just a meat sack and we don't have free will and there's no culpability. And how could there be a God that would make us be culpable since we're just a directionless force in nature? In this article, it says the current return of the organism and the recent calls for a genteel perspective in evolutionary biology. And what they mean by that is what's happening as a result of epigenetics is that they're using the word organism as agent. Okay, it's we're no longer just being the results of played DNA, but the organism controls the DNA because it has all these epigenetic overlays. So the way to look at everything is as the cell is an agent and the overall organism is an agent, and get away from what we, you know, from the gene-centered view of everything. As a result of this perspective in evolution, in evolutionary biology, they point out that we still have some thinking to do considering this side of Kant's legacy, and that's referring to teleology, which Kant argued for. And the name of this article is Teleology and the Organism, Kant's Controversial Legacy for Contemporary Biology. So basically, it's being argued more and more that teleology or design is back on the table. With random mutations in retreat, scientists have proposed that natural selection works at the RNA instead. You know, it was supposed to be up at the DNA level, but now they're arguing it's at the RNA level. And this is a picture of a mechanism which was developed 30 years ago called the selection mutation drift theory. What people were realizing that codons, which we talked about last time, are not random, but they're biased. And that pretty much did away with random mutations at the same time. And they were trying to figure out how could these codons, which seem to have a second code in the DNA, how could they work by natural selection? On the far left in the middle, they talk about, you know, optimal codons can speed up translation of the RNA to a protein. You see the little ribosomes in blue coding out proteins, and they can make it, they can change it from slow to fast. And more optimal codons make it go faster. And they say, well, that variation may be natural selection because there's still some variation in it. And if you go down to the bottom and then go counterclockwise, you see the two X's, which are chromosomes. They say that this is fixed by selection, but they're really having a hard time doing that because they're basically, at the same time, they're saying that protein and RNA is fixing the DNA. Now, epigenetics says that, but supposedly neo-Darwinism doesn't say that. So how can you say that? Because you're going backwards up the central dogma tree, aren't you? Recall that the central dogma is a one-way direction of information from DNA to protein to RNA to protein, and that it doesn't go backwards. But they are in essence happen to, X their own thoughts, throw central dogma under the table to somehow come up with a way for this fixation to go back into the synonymous codons there. Now, other scientists say that these regions, far from contributing to human adaptation, which is natural selection, might represent the Achilles heel of our genome. I mean, that's basically, the Achilles heel. This is going to make or break the whole everything, really, because it stands to do away with natural selection. Subsequently, they call it the Achilles heel. In it, they discuss positive correlations between genetic diversity and recombination do not necessarily imply that selection is affecting diversity in the genome. And this is discussing the effects of GC bias, and that GCs are those two nucleotides that are involved very much in epigenetics. And the patterns of these changes that occur follow along a biased, non-random fashion. So it does away with this model, basically. They're saying there's still natural selection in the works. We just got to prove it, but this is losing out. This is natural selection's last hope, if you will. Now, here's a couple of long-term things I want to talk about. The cytosine nucleotide on the far left, it's the thing that gets methylated by the epigenetic enzymes. And when you have a 5-methyl cytosine where the methyl group is on it, it spontaneously will deaminate. In other words, it'll come off and it'll form a thymine. And that occurs according to a specific pattern. So it's not really random. It's a normal pattern that was a teleological pattern built into the DNA that this would tend to just wear down from the C down to the T. And so on the long view of organisms over even hundreds of thousands of years, there's this trend towards going from a cytosine down to a thymine, but it's due to the epigenetic overlay. At the same time, there in the repair mechanism during duplication of the DNA, there's this tendency to go from AT to GC, and that's called the GC biased, which that slide before talked about. There is no natural selection built into this. This is just the cells doing what they do. And that's what has gotten a lot of those guys quite upset. They don't like the fact that this might explain all the codon biases in the DNA, and it is being proved that GC bias causes codon bias, and codon bias is that second code that we discussed in the last talk. As well, there's another mechanism that's occurring. This is on the left side. You see the blue, what looks like a comb, and that's the RNA, and it's matching up to transfer RNAs, which you see in light blue, E, P, and A. And these transfer RNAs are adding amino acids on one at a time. We discussed this last time. But if you see there's a GAG and the CUU letters, those the GC in a that is circled in black is the wobble pair. And we're not going to really go into the full aspects of the wobble pair, but the wobble is it's a section of DNA pairing that is not under the same Watson-Crick rule, but it has basically a wobble to it. So there's a little play in that mechanism, but that play in the mechanism tends to favor GC letters or acts better on it. So it's also adding to the epigenetic GC bias. So instead of this mechanism that says that, well, the slow and fast translation is causing natural selection, we have this mechanism which is a cellular mechanism, a cellular teleological mechanism that is built to do this. And then we have this mechanism, which is the same without natural selection. In fact, they say that natural selection, that the third codon position or the wobble position is immune to natural selection, basically. So it's important to understand that epigenetics on day-to-day actions are much faster than epigenetic bias mutations, which occur over vast time. They can markedly increase in speed due to the junk DNA, though. We know junk DNA is also called the jumping genes. During radical environmental changes, these changes can occur extremely fast. This might even explain Gould's punctuated equilibrium. I'm not sure if you're aware of that, but Gould broke away from the neo-Darwinists when, I think he was a paleontologist, and he just realized that the Cambrian and other extinction events could not be explained by the gradual motions of neo-Darwinism. And he broke away from them and came up with the concept of punctuated equilibrium, that after major extinction events, that things change very rapidly. Of course, he had no idea why. But the talk amongst biologists and geneticists is that maybe this is going to be due to junk DNA, because it's the only thing that can do it, probably. What's also interesting is Gould brought up the concept of the evolutionary clock of time. If you took everything back and you wound the clock back and re-released it, you should come up with completely different things according to neo-Darwinism and evolution. So this is a very debated topic, and they've long debated whether rewinding the tape of life would give the similar results, or whether outcomes depend largely on chance events that push the course of evolution into radically different tracks. This is off of an article called "The Tape of Life May Not Be Random." In this article, they tied basically epigenetics to show that epigenetics will tend to push things into one particular type of formation for an organism. And so the ultimate cause of these patterns is not natural selection. I mean, that's a slap down right there, but rather a strong phenotype basis in the RNA genotype phenotype map, a type of developmental biases of findability constraint, which limits the evolutionary dynamics to a hugely reduced subset of structures that are easy to quote unquote find. And the article is called Phenotype Basis, Phenotype Bias Determines How Natural RNA Structures Occupy the morphospace of all possible shapes. So in other words, the overlay of these long, slow epigenetic changes and fast epigenetic changes tend to cause things to converge. And we've noticed in biology that we would see, for instance, the development of eyes 100 times over the history of life. Well, how could that occur? How could you ever get an eye hardly the first time, much less 100 times? So this concept of convergence, they just said, well, it's just neo-Darwinism. But this, on the other hand, explains how the same types of things tend to come out. Now, this is from the Psalms, and RTB quotes this as a part of their progressive creationist concepts. They say all the creatures wait for you to give them their food in their due season. You give it to them. They gather it up. You open your hand, and they're satisfied with good. You hide your face, and that would be, say a meteor hitting, and they're dismayed. You take away their spirit, and they expire and return to the dust. And then you send forth your spirit, and then they are created and renew the face of the ground. This is a very RTP has commented that this looks a lot like what's happened during the Cambrian and other periods where things suddenly are destroyed and then they're recreated. We don't know how it's recreated or how the Holy Spirit did it. It could be you rearrange the DNA that's there and it forms particular forms that typically tend to happen. 4 legs, tails, etc. There might be different sizes, and this all may be wrapped up in our junk DNA and epigenetics. So this is kind of a little diagram showing epigenetics on top of the DNA and how it can form different forms. And the different epigenetic tags can form different cell types listed below. This is kind of an interesting application. Epigenetics, the making of an ant cast. There was a huge argument with Richard Dawkins and E.O. Wilson. One of the guys here in Montgomery knew E.O. Wilson because he grew up in Montgomery. E.O. Wilson was one of the great names in evolution. I think he was at Harvard. And E.O. Wilson was on Marxist, and he, he was, I think he was an entomologist as well. And he said to Dawkins, how can you have the same DNA in bees and ants, and yet they can have all these different forms? It just doesn't, neo-Darwinism and mutations doesn't explain all that. But epigenetics does. Social insects represent a unique model for how the entire genome can give rise to different phenotypes. You can get soldiers, common laborers, and queens. New research on ants and honeybees bees point to the DNA methylation, which is epigenetic, Lamarckian epigenetics, as a crucial factor in determining the caste. Now, this diagram on the top was the original diagram that Conrad Waddington made, and it really gives a good concept of what he's doing. You got the marble on the top of the middle when you're at in your embryological stage, and then as you develop, the marble rolls down and goes through different paths to form different overall beings. And on the far left, you have the minor ant. In the middle, the soldier. On the far right, we have the queen. And it's all due to that marble taking the different epigenetic pathways. Now, one of the interesting things about this is when you look at the ants, the soldier ant, which is like the soldier, the soldier bee, like in the middle, it makes the royal jelly. And the royal jelly is what they feed to the pupa of one of the developing bees, and it turns into the queen bee because it gets this jelly. So this means that the environment changes that queen, and it takes the part of the epigenetics to make it into a queen. But since it's royal jelly and made by the soldier, how in the world did they know to do that? It was in their genetics and epigenetics. It's kind of an irreducibly complex thing if you think about it. Because you can't have the soldier without the queen, and queen can't make the soldier without the royal jelly that the soldier makes. So now Darwin's finches that's the key to his theory of evolution. When he was on the Galapagos Islands and he saw the different finches on the left with different beak sizes, he reasoned that the conditions of existence changed those beaks so they had variation and they passed that variation on. There's been 19,000 articles on it because everyone kind of goes back to the Galapagos Islands. It's kind of like going to Mecca, doing the 7 trips around it. That's kind of like, and so they've done, and not a one of those 19,000 research trials really could demonstrate how mutations change those beaks correctly. But now with epigenetics showing up, I'm going to show you a study in the next slide. I'll go ahead and do it now. Michael Skinner, he came up with this study where he studied he compared the epigenetic changes amongst the different finches, and it said the epigenetic changes guides the molecular basis of the evolution of the finch. The DNA is still there, but the epigenetics played them differently to make the different beaks. And when he compared it to the point mutations, which neo-Darwinists study, the point mutations didn't line up like the epigenetic changes did. So the point mutations basically didn't account for the beak change in the finches, but epigenetics did. I mean, that's huge. This is the icon of evolution, this example, and it's been upended by this gentleman. Going back to junk DNA, on your far left, we have Barbara McClintock. And now she was doing her research in the 40s, right? This is right around the time that the modern synthesis, and as well as Conrad and Waddington, came out with their statements. And have you ever been to, well, everyone's seen corn, natural corn that has different colors of kernels, and they're all different all over the place. Well, she said why are they different? Why aren't they all the same? Why aren't they all the same yellow or other things? And why do they seem to be in one part and not the other? So she started studying the DNA, and she noticed that the genes, the junk DNA genes, were jumping about. She called them jumping genes, and that they were turning certain things on, like color. So one kernel might be dark and the other one might be golden. And through her, what's fascinating about this is there were some people that went to visit her in her lab because they'd heard of this stuff that she was doing. And they basically said she's either crazy or brilliant. And they pretty much concluded she was crazy because her stuff was not inconsistent, was not consistent with the theory of the day. And she didn't care about the theory of the day. She just did her studies and told it the way it was. Okay, so she gets overlooked, right? And then the Human Genome Project comes out in 2001, and it was a complete flop because they ignored her junk DNA, basically. And right afterwards, the Nobel Committee said, holy cow, this lady discovered junk DNA, and we overlooked her all these years. So there she is at 83 years of age, getting the Nobel Prize when she probably should have gotten it, 50 years earlier and changing the course of genetics and biology. We talked about this last time. There was the ENCODE project came out after the Human Genome Project didn't hit anything. And there were thousands of folks in it, and they pretty much wrote the eulogy for junk DNA. And they concluded that maybe upwards of 95% of this junk DNA is actually functional. This is an article that came out by a cardiologist researcher. And he said medical researchers ignored evolution's so-called junk DNA. Since then, researchers have been exploring these possible functioning non-coding transcripts. The discovery of functional small peptides translated from the junk DNA has encouraged researchers to re-examine its rules. They re-examined it. They'd ignored it, but now they re-examine it. For example, junk DNA has been associated with cardiovascular disease. So-called junk DNA plays critical role. The title of this is so-called junk DNA plays critical roles in mammalian development. So there it is. The heart disease, which kills over half of all the deaths or heart attacks, we were looking at the wrong stuff. In addition, cancer, which is the second leading cause of death, may depend on understanding epigenetics. And I say may. There's like 40,000 articles that say epigenetics is involved with cancer. So it's more than a may, I think. Let me see. Okay, so what is this doing, the epigenetics, and how is it influencing everyone? And I think I showed this at the last talk, but this was in the Federalist, and they said the field of epigenetics causing a major disruption in the field of evolution. They said that current estimates are that approximately 1/3 professional academicians who do not believe in intelligent design find Darwin's theories inadequate to describe all the complexity in biology. Now, one of the questioners last time pointed out, well, yeah, there's still 65%. However, this 1/3, these were the people that believed evolution was a fact like gravity. To have, I would say, even if 5% moved, it would have been significant. But we have, a huge chunk of people that have moved. And I would say that this is going faster and faster because this is a little bit of an older article. I also quoted this at the last talk, and this was a Royal Academy meeting at Cambridge, I want to say. And it was a new way of scientists argued that mainstream evolutionary theory needs an urgent overhaul. This Royal Academy meeting, by the way, it was started by a Nobel Prize guy that was the head of the meeting. And when he announced that he was going to study epigenetics and its influence on evolution, there were at least 20 notable people that wrote articles, letters of disgust saying, no, we shouldn't pay attention to it at all. And the guy couldn't believe it, because he said, look, we'll see their point of view. So they eventually did it. And a lot of people went there. And one of the persons that was for the discussion of epigenetics, and I can never pronounce his name, was Pigliucci. He gave a talk on it basically, and somebody asked him during his talk that isn't this disagreement between the EES proponents, that's an extended evolution, which includes epigenetics, And the more conservative biologists, which are the neo-Darwinists, certainly looks more like a culture war than a scientific disagreement. And he said, sure, it's a culture war, and we're going to win it. And half the room burst out cheering. Now, I submit that half is greater than a third, but I would also submit that that half was probably the younger geneticists, because if you look on, you look at the growth in the different biology programs on the universities, epigenetics is outstripping everybody else. I mean, they've had 120,000 journal articles published in the last 10 years. It's where it's at, you know. So as if you can tie your research into cancer and heart disease, you're going to get money. So that's why they're going that way. It's not like they're philosophically love the idea necessarily, but they want to do it for the job. This is that article that talked about that talk. It's in the Guardian, and it says, "Do we need a new theory of evolution?" So I start off with a 9-year-old one from Nature saying, "Do we need a evolution need a rethink?" And now again, they're trumpeting, "Do we need a new theory?" But this is a great article, by the way. It's a very long article. I mean, it's a long read, but it really explains the whole history of what's been going on. This is an article by, or a quote by Dennis Noble. With neo-Darwinism's replacement by epigenetics, teleology is back on the table. I mean, that's a remarkable quote from a guy that doesn't believe it. He's not a creationist. And this is in an article he wrote, "Was the watchmaker blind, or did she have one eye?" And that's a tongue-in-cheek poke at Richard Dawkins and his Blind Watchmaker book, you know, who says it's just DNA blindly. But Noble is pointing out that she's got one eye at least. So and teleology is back on the table. So this is an overview then, basically, of what we stand to talk about. Epigenetics is in the middle of it all. And you see DNA is a small portion in the bottom to the right. And then you see the genes and nucleosomes and all these other concepts, but they're all wrapped in to epigenetics as being the key to it and explaining everything. So anyway, for more information on this design concept that we're getting into, now, I don't think Hugh talks a lot about epigenetics, but he wrote his book called Design to the Core, which does look at it from a teleological design method. And of course, you can email me if you want. Also, you can find me on LinkedIn, YouTube, on Darwin versus Lamarck Facebook page. Is Kirby Hansen here tonight?
Yes, Kirby's here.
I am.
Oh, you are? Okay. This next slide's for you. Kirby has helped me a great deal with my slides. And so I had to pretty much throw a third of them away and start new. but if anyone else can jump in there and really hit me hard, I don't want this to just a sound of one hand clapping. I like the heavy criticism. So that one's for you, Kirby. So you want to break out for questions?
Okay, now we open up for question. Let me pause the recording